Alright, I'm back, and this is the next part of Chapter 10. Section 10.2. This chapter is called RNA. I have not read this yet, and this is more the part that I'm confused with, so please bear with me if I aren't able to explain it right. But I'm going to try my best. Okay. RNA. Recall that the nucleotides in DNA molecules are grouped into genes that contain the information needed to make specific proteins. In eukaryotes, the genes directing protein production are in the nucleus, the enzymes and the amino acid building blocks for protein production in the cytosol. So, amino acids are the building blocks for protein. So, that's why, like, muscle milk and stuff says we have amino acids, because amino acids make up protein. The nucleotide acid called ribonucleic, okay, I think I got that right, ribonucleic acid, RNA, is responsible for the movement of genetic information from the DNA in the nucleus to the site of protein synthesis and the cytosol. Structure of RNA, sorry. A lot of new vocab in this section, so be prepared. Okay. Like DNA, RNA is a nucleic acid made up of repeating nucleotides. However, as figure 10.6 shows, RNA differs from DNA in its structure. The sugar molecule of every RNA nucleotide is ribose, whereas DNA nucleotides contain deoxyribose sugar. The name ribonucleic acid is derived from the name of its sugar, just as the name deoxyribonucleic acid, okay, I never said I would be perfect at pronouncing, I apologize, is derived from the name of the sugar. A second difference between RNA and DNA nucleotides is that thymine is rarely a part of RNA molecules. Alright, here comes the big one. Gotta pay attention to this part. Uracil. And yeah, that part confused me. Uracil. Uracil, a nitrogen-containing pyrimidine base, usually replaces thymine in RNA. So from that, you're probably thinking, nitrogen-containing pyrimidine base? What you need to know is uracil is part of DNA, and it takes the place of thymine. So in DNA you have thymine, in RNA you have uracil. It's very basic. I got taken away from all the fancy wording that they had. Alright. As a result, uracil, not thymine, pairs with adenine in RNA. So instead of my acronym I used to remember adenine and thymine, I say it's AT. Now you have A and U. So if you have a U-based nucleotide, then you're going to pair it with an adenine, and an adenine is not going to be paired with a thymine anymore. It's going to be a uracil. They have a picture of it, but it's just a picture of DNA. It's not really that helpful. Types of RNA. Whew, this is the complex part. Alright. RNA in 3 different types. Each type of RNA has different functions. The 3 types of RNA and their common abbreviations are as follows. Messenger RNA, which is often referred to as mRNA. M is lowercase, RNA is capital. mRNA consists of RNA nucleotides in the form of a single uncoiled chain. mRNA carries genetic information from the DNA in the nucleus to the cytosol of the eukaryote cell. Okay, so messenger RNA pretty much says the whole point of this. mRNA, it takes information from the DNA and it takes it outside of the DNA, or of the nucleus, which is the cytosol. If you don't know what the cytosol is, it's the area in between the nuclear membrane and the nucleus. So it's all the jelly-like stuff in between. So messenger basically is a messenger. It takes the information from the nucleus and takes it out to the cytosol. Not that hard. Transfer RNA, tRNA. tRNA consists of a single chain of about 80 RNA nucleotides folded into a hairpin shape that binds to specific amino acids. There are about 45 varieties of tRNA. So, consists of about 80 RNA nucleotides. Ooh. So, transfer RNA is basically, I believe it's a bundle of nucleotides, and it's in this hairpin shape, and it binds to certain amino acids. So it takes the nucleotides to the amino acids, and that's how it transfers. I'm not sure exactly how that goes, but all you need to remember right now is that it's a bundle of nucleotides in a hairpin shape that binds to amino acids. I have trouble understanding some of this, so bear with me. The third form is ribosomal RNA, rRNA. That's not confusing. rRNA is the most abundant form of RNA, so this is the most common, and usually ribosomal RNA is just like there. rRNA consists of RNA nucleotides in a globular form joined by proteins. rRNA makes up the ribosomes where proteins are made. So rRNA is the most common form. It's made up of a bunch of nucleotides in a big glob. So you have the thymine, guanine, adenine, all of that, and they're just jammed together in this big glob. They are joined by proteins, so it's the nucleotides and the proteins in this big glob, and it makes up the ribosomes where the proteins are made. I guess that's just how they put it. I hope I'm not the only one that's confused by this, but transcription. One function of RNA is to carry genetic information from DNA in the nucleus to the cytosol, where it can be used to produce proteins. The process by which genetic information is copied from DNA to RNA is called transcription. Transcription is a big part of this whole process, so I'm just going to repeat that again. The process by which genetic information is copied from DNA to RNA is called transcription. So basically, all of these nucleotides that make up the gene they're synthesized by transcription, and transcription takes these DNA forms and turns them into RNA. So if it's, for example, like AUG, it would be transformed to uracil wouldn't be in a DNA, I'm sorry. So it would be like ATG. So if it's ATG, then it would be transcribed to A would turn into T, but T is replaced by uracil, so it would be AGC. Okay, so G would turn into cytosine, and cytosine would turn into guanine. So that would be transcription. Okay, this is all I have for this part of it, and if you have any corrections, I'd love for you to put them down, because I hope I'm not completely screwing you guys up on how this works. So yeah, thanks.